Hello everyone, today we'll talk about UPHESC Zoology 2022 previous year question paper. So today we'll discuss the part 2. This paper has two parts. The part 1 was GK paper and it has 30 questions, and the part 2 was these optional subject and it has 70 questions. Now, this question number 1 in the optional subject. So, this among the following, which group of organism is not uricotalic? So, answer is mammals. Mammals are they excrete urea. So, the mammals are ureotalic in nature, whereas the birds, insect, and reptiles they are uricotalic. So, in this question, it is asked which organism is not uricotalic. So, answer will be mammals. Next question is kidney of men is pronephros, B metanephros, opisthonephros or mesonephros. So correct answer is B metanephros. Which hormone is a modified amino acid? So prostaglandin, estrogen, progesterone or epinephrine. So prostaglandin, estrogen and progesterone they all are steroid hormones derived from lipid. Epinephrine is an amine hormone derived from the amino acid tyrosine. So the correct option is C, epinephrine. Next question is receptor for FSH is present on A. Leydig cells, B. Sertoli cells, peritubular cells or spermatogonium germ cells. So FSH, the receptor for FSH is present on Sertoli cells. So FSH, it is secreted by anterior pituitary. And then in testes it acts on Sertoli cells. Next question is estrogen and testosterone bind to. So these both estrogen and testosterone they are steroid hormone. They are lipophilic or hydrophobic hormone. So they are lipophilic in nature, lipophilic, which means that they can cross the lipid bilayer. So they can cross the lipid bilayer, and that's why their receptor are present in the cytoplasm. So they bind to cytoplasmic receptors. Unlike the hydrophilic molecule, whose receptor are present on the membranes. Examples are G protein coupled receptor or enzyme link membrane receptors. So in that case, whose ligand are hydrophilic in nature, and if they are unable to cross the plasma membrane, their receptor lie on the cell membrane. Next question is quick response towards stress is done by androgen, corticosteroid, steroid or epinephrine. The correct answer is epinephrine. Epinephrine also known as the adrenaline hormone. Adrenaline or 3F hormone. Fight, flight or fright. So if it works under different kind of stress, anxiety, if there is an injury, in all those cases epinephrine works. So this is the stress hormone and it is secreted by the adrenal gland. Now next question is genetic recombination is due to option A independent assortment, option B linkage, option C mutation and option D incomplete dominance. So genetic recombination happen due to independent assortment of chromosome during prophase 1. So in that the chromosome from the paternal side and the maternal side they assort independent of each other, which leads to genetic recombination, that is formation of the new combinations. Next question is pseudogenes are the results of. Answer is unequal crossing options are unequal crossing over, gene duplication and mutation events, or mitosis. So pseudogenes are the result of gene duplication and mutation events. Next question is when heterozygous offspring is crossed with the recessive parent, it is called as test cross, reciprocal cross, monohybrid cross, or the dihybrid cross. So it is the correct answer is test cross. So Monohybrid cross means when only one trait is considered. For example, the height, like this. And dihybrid cross is when two traits are considered for the cross, like height and the shape of the seed, like this. So when two traits are considered, then it is a dihybrid cross, and if only one is considered, then it is a monohybrid cross. Reciprocal cross is one when the genotype of the parent is changed. For example, if in the first cross the father male is tall and female is small, so in the reciprocal cross their traits will be exchanged. So in the reciprocal cross, the father will be now small t, and the mother will be taken as the capital T, capital T. This is when the genotype of the parent is reversed. Test cross is when the when the offspring, let's say this is the cross and this will be the offspring. So when this offspring is crossed back with the recessive parent, it could be either male or female. So when it is crossed with the recessive parent, then it is called as the test cross. So answer is A, test cross. Next question is aneuploidy with chromosome number 2 and minus 2. 2 and minus 2 means both the copies of a chromosome are missing. So it will lead to, there will be 0 copies of a particular chromosome. So it will lead to nullisomic. And monosomic means there is only one copy, 2 and minus 1. It means one copy is missing of a chromosome. Trisomy is when there are 3 copies of a chromosome. So it will be 2 and plus 1. Tetrasomy means there are 4 copies, 2 and plus 2. So the correct answer will be C, nullisomic. Next question is hemophilia and the royal families of Europe is linked to autosomal recessive trait, X-linked recessive trait, X-linked dominant trait, and Y-linked dominant trait. So in royal families, it is the family of Queen Victoria, in which she was the carrier of the hemophilia, and she passed on this to her offsprings, and that's why it is also called as a royal disease, and it is an X-linked recessive trait. Which among the following group is known to have cellular grade organization of the body? So protozoa, they are single cell eukaryotic organism. Annelida, they have tissue grade of organization, and metazoa belongs to all animals. So porifera, they have cellular grade of organization. So in porifera, there are no tissues, only cells arranged to form functions. There is some certain sort of division of labor in porifera. Which among the following is an anthrocele animal? So if we talk about coelomic organism, coelom is of two type, schizocoelom or the organism are either schizocelic or anthrocelic. Anthrocelic. So the classification is based on the origin of coelom. So schizocelic this tinea is a platyhelminths. So they are acoelomate. There is no coelom is present. SK elements, they are pseudocoelom. There is no true coelom is present. And from the Annelida onwards, we see the true coelom. So Annelida, arthropod, mollusk, they have the schizocoelom. Annelida, arthropod, and mollusca. Whereas the anthrocoelic, these echinoderms, echinodermata, hemichordata, and the chordata, they have anthrocoelom. They are anthrocoelic in nature. So Asterias is an echinoderm, and in this question it is asked which is an anthrocoelic. So Asterias, it is an echinoderm, and that's why the answer is Asterias. Ferritima is earthworm. Annelida, so Annelida has schizocoelic. Tinea platyhelminths, they are acoelomate, and Pelimon arthropod, Pelimon is an arthropod. Pelemon is prawn, so it is an arthropod, and the coelom is caesocelic in nature. So correct answer will be A, Asterias. Basis of classification of phylum is, the answer is uh, shell. Next question is, which of from which subclass uh, it is either on the basis of shell or the type of radula. Next question is, from which subclass of class Reptilia have the mammals evolved? So options are Anapsida, Eurypsida, Diapsida, or Synapsida. So correct answer is Synapsida. Next question is, which one of the following is converted into thyroid gland of vertebrates? A. Dorsal lamina, B. Endostyle, C. Epipharyngeal group, or D. Neural complex. Correct answer is B. Endostyle. Which of the following is the reptilian character of monotremes? Body covered with hair, pinnae present, dicondylic skull, or the T-shaped interclavicle. So answer is D. T-shaped interclavicle. Next question is, genes having exons and introns are termed as option A. Pseudogenes, B. Split genes, C. Overlapping genes, or D. Homeotic genes. Correct answer is B. Split genes. Animal those have a foreign gene deliberately inserted its genome is, answer is A. Option is A. Clone, B. Hybrid, C. Transgenic, or both A and C. The correct answer is transgenic. Which factor is least responsible for genetic drift? Option A. Migration, founder effect, bottleneck effect, or the option D. Restriction of resources. So correct answer is D. Restriction of resources. Which of the following is characteristic of allopatrid speciation? Options are dense population, asexually reproducing population, geographical isolation, or adaptation of alleles. So, allopatric speciation is due to geographical isolation. Next question is, in which of the following period of Paleozoic era the first vertebrate appeared? The first vertebrates were fishes. So, they appeared in the Ordovician period. Hardy-Weinberg's law give concept of option A mutation, option B genetic drift, option C natural selection, or D genetic equilibrium. So Hardy-Weinberg gave the law genetic equilibrium of a population. The change of behavior by life experiences is called as A instinct, B maturation, C imprinting, or D learning. So the right answer is learning. So learning affects our behavior, and we get this by life experience. Which of the following is not an attribute of population following Hardy-Weinberg law? So those population which follow Hardy-Weinberg law, so there should be large population size, and there should be absence of evolutionary force, and there should be random mating. There should be mating should be random, and there should be no migration. So there should be no influence of other out, outer resources. So in this, the correct answer will be non-random mating. This is in this question it is asked not an attribute. So non-random mating. So which is biased mating. So this is not an attribute of Hardy-Weinberg law. In mating should be random. So correct answer will be non C. Gastrulation in vertebrate results in the formation of option A blastocoel, option B three germ layers, option C archenteron, option D archenteron and germinal layer. So correct answer is D, archenteron and the germinal layer. Next question is, the vertical migration of plankton is an instance of, answer is circadian rhythm, um, photoperiodism, and photokinesis. All these three are correct. Circannual rhythm, this is not correct. All these three are correct. Next question is, during ecological succession, one of the following takes place. Number and types of animal remain constant. It is false. there is change in the number and the type of species occupying a space. The change leads to a community that is in near equilibrium with the environment. That is right. Gradual and predictable change in the species composition occurs in a given area. So according to commission, this was the right answer. This was also right, but according to me, it is not right because the change is not predictable. So it should be wrong, but the commission has taken this answer as right. Both B and C as right. Establishment of new community in a very fast and in its private phase, this is wrong. It is a very slow process. The enzyme which catalyzes the first step of glycolysis is, so it is glucokinase and hexokinase, both catalyze. So it depends upon the tissue which kinase will be acting. So because in this no tissue has been specified, so that's why both A and B option will be correct. Next question, the carbon dioxide is the primary product of. So it is a Krebs cycle where the two CO2 molecules are released. Okay. So in glycolysis, the 6 carbon compound glucose is converted to 3 carbon 2 pyruvate molecules. So there is no release of CO2. As similarly in ETC, electron transport chain or the phosphorylation, there is just movement of electron and generation of oxygen. So in all these, there's no release of CO2. It is a Krebs cycle. In one Krebs cycle, two CO2 molecules are lost. Nicotine adenine dinucleotide phosphate is generated in, so NADPH. It is synthesized only and only in pentose phosphate pathway. Sertoli cells are found in, so Sertoli cell found, we have already talked about this. Sertoli cell are found in testes, and they have an important role in spermatogenesis. Which of the following is true for microRNA? It includes all tiny RNAs. It is produced as a large RNA. It acts as a transcription factor, or it regulates gene expression. So the correct answer is D. It regulates gene expression. All these are incorrect. Next question is, allosteric inhibition of an enzyme involves cooperative binding of a substrate to an enzyme. False. Binding of a negative modulator competitively to substrate site. No. And binding of a negative modulator non-competitively to a substrate binding sites. No. It knows binding of a negative modulator to a site other than the substrate site. So the correct answer is D. Next question is, the catalytic efficiency of an enzyme is represented by Vmax plus Km, Km, or Kcat, Kcat by Km. Correct answer is Kcat by Km. So the catalytic efficiency of an enzyme is determined by this Kcat by Km. Km is the substrate affinity. Transmembrane domain of protein are typically beta sheets, alpha helical, random coils, or unstructured though. So the right answer is alpha helical. The transmembrane region which pass through the transmembrane are right-handed alpha helix in nature. So next question is, Golgi apparatus is concerned with. So options are secretion, excretion, ATP synthesis, or RNA synthesis. So Golgi apparatus is concerned with the secretion of proteins. So proteins which are synthesized in ER are transported to Golgi, and then Golgi act as a sorting station for plasma membrane or for the intracellular organelles, lysozymes, or the peroxisome, or mitochondria, wherever whatever the intercellular organelle, or to the outer surface. So it has a role in secretion. Many glycosylation modification like Olin glycosylation, addition of glycosylation, all these takes place in the Golgi. The composition of a nucleoside is, so base plus sugar, base plus phosphate, base, sugar, and phosphate, or none of these. So in case of a nucleotide, there is a sugar ribose or deoxyribose. To this sugar is attached a base, and to this sugar there is a phosphate group attached. So this base and sugar, this is called as nucleoside, and this whole sugar, this nucleoside plus phosphate is known as nucleotide. The correct answer will be A, base and sugar. Which organelle produces spindle fiber during division of an animal's cell? Chromatin is a DNA. Vacuole has no role. Mitochondria does not have role in spindle formation. It is the centriole which uh, divides at the time of cell division and then moves to the other pole. And then there is a generation of these microtubules during the mitosis. Now, an environmental agent that triggers transcription from an operon is depressor, repressor, controller, or inducer. The correct answer is inducer, which induces gene transcription from an operon. The mitochondrial DNA is single-stranded circular, single-stranded linear, double-stranded linear, or double-stranded circular. The mitochondrial DNA, mitochondria are, there is a theory of symbiosis which says that the mitochondrial DNA or mitochondria have originated from the bacteria. So their genomic DNA resemble those of bacteria. So the answer is it is double-stranded and circular, like that of bacteria. Next question is synaptonemal complex is formed during Pachytene stage, Diplotin stage, Dicotin stage, or the Zygotin stage. So synaptonemal complex when the two homologous chromosomes they pair up, when the pairing of the homologous chromosome take place. So it forms synaptonemal complex during the prophase 1. So it forms during the prophase 1 of the zygotin stage. Which of the following is not a cell death mechanism? Apoptosis, necrosis, and autophagy. All these three are cell death mechanism of a cell. Endocytosis is a process of uptaking material or like receptor uptaking from the cell. So when it is like this, and then this will be engulfed, and when something is engulfed from outside, there will be some vesicle formation, and then it will be endocytosis inside the cell. In TEM study, double fixation is done with. Double fixation means there are two fixation. One first primary with the with an aldehyde, so glyceraldehyde or formaldehyde, and then with the secondary fixation is with the osmium tetroxide. So in this, all these options are correct. This question was deleted by the commission. In which phase the cell cycle DNA replication occurs? So prophase, anaphase, telophase, or interphase. So DNA replication takes place during S phase, and before this there was a G naught phase, that is called G phase, and the gap 1 phase. And during the prophase, so DNA replication happens during the S phase of the interphase, whereas during the M phase, which consists of the prophase, metaphase, anaphase, and telophase, there is just the division of these chromosomes happen. So answer is A. Answer is B, interphase. Next question is DNA replication in eukaryote differ from prokaryotes in being unidirectional rather than bidirectional. It is false. It is bidirectional both. Being discontinuous rather than continuous. It is also false. Using DNA primer instead of RNA primer, this is also wrong. RNA primer is used in both. Having different enzymes for loading and lagging strands. So this is true statement. In prokaryote and eukaryote, the enzymes used for replication are different. Among the following eukaryotic polymerase, which one is not associated with duplication? DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, and DNA polymerase epsilon. So answer is DNA polymerase beta. EF2 or protein synthesis is called as. So EF2 is called as translocase. Answer is A. Prokaryotic RNA polymerase. Okay, so other options are transformylase, lysyl hydrolase, or peptidyl transferase. The correct answer is translocase. Next question is prokaryotic RNA polymerase is inhibited by penicillin. Penicillin is a cell wall inhibitor, antitoxin, general term. Alpha-amanitin, it is an ETC inhibitor, and rifampicin. So correct answer is rifampicin is a prokaryotic RNA polymerase inhibitor. Leader sequence in mRNA of eukaryote is found in. Options are within the first exon, between the transcription and translation site. This is the correct option. After stop codon, no. In between exon and intron boundaries, no. ELISA is used to. So ELISA is enzyme linked immunosorbent assay. It is used to detect antigen or some antibody also. So in this case, the protein is identified. It is used to isolate DNA, no. It is used to purify protein, no. Separate viral RNA, no. It is used to identify specific protein that we call as antigen or antibody. For example, for detection of HIV antigen, for corona detection. So in that detection, the antigen on those pathogens are detected with the help of an antibody. Positron emission tomography detects electrons, neutrons, positron, or photons. The correct answer is photons, not positron. Chargaff rules is A plus 2T is equals to G plus C. So we know, even if you don't know the Chargaff rule, we know that A is bonded with T and G is triple bonded with C. So A plus G will always be equals to T plus C. So this is the Chargaff rule. A plus G will be equal to T plus C. These are not the right statement. This could not be the option. A plus 2 T plus is equals to T plus 2. This is also a false statement. The technique used to inactivate a gene by altering the DNA is antibody microinjection. Wrong. So antibody is a protein molecule, so you cannot alter a DNA. Antisense nucleic acid block, no. By homologous recombination, this is the right option. So in this case, for example, this is the native gene, and here we can have some marker like this or some non-specific sequence. This sequence will be homologous. So here at this place, homologous recombination will take place and this will be exchanged. So homologous recombination leads to alteration of the genetic component. And other option is introduction of dominant inhibitory mutants. The next question is, which enzyme act as a biological scissors in genetic engineering? Ligases, nucleases, polymerases, or restriction enzyme. Ligase is used for biological scissor means something that is used to cut DNA or RNA. So ligase, they join. Ligase are used to join two pieces of DNA. Nuclease, it degrades nucleic acid. Polymerase, it amplifies the DNA. Restriction enzyme, so there are three types of restriction enzyme: type 1, type 2, and type 3. So type 2 restriction enzymes are used in genetic engineering recombination because they cleave within, if this is the restriction site, they cleave within the restriction site. So type 2 restriction enzyme are used in the genetic engineering. And restriction enzyme are called as the biological scissors. For in vivo gene therapy, genes are usually delivered through electroporation, liposuction, adenoviral vector, or microinjection. So in case of animal, adenoviral vector, because it is a DNA virus, and liposuction, both these methods are used for gene transfer. CD19 marker is a marker of B cells, T cells, macrophages, natural killer cells. The correct answer is A, B cells. Next question is, which of the following blood cells is involved in antibody production? Option A, RBCs, B, T lymphocytes, C, B lymphocytes, and D, neutrophils. So in antibody production, B cells are involved. Only B cells are are those cells, immune cells in our body, which are able to produce antibodies. So B cells are used in the hybridoma technology for producing the monoclonal antibodies by fusing the B cells with the cancerous cell line. The most abundant okay, it is asked in the blood actually. So it is B lymphocyte. Only B cells produce antibody in our body. The most abundant immunoglobulin is IgA, IgE, IgM, or IgG. The most abundant is Ig. In a cancer patient, the defective gene can be determined by using eastern blot, northern blot, or western blot, or southern blot. So for DNA, we use southern blot. For protein detection, western blot is used, and for RNA, northern blot is used. Receptor for signaling of steroid hormone are located at. So we have already talked about it. So the steroid hormones, they can cross because they are lipophilic in nature, they can cross this plasma membrane. That's why these receptor are located inside the cell cytoplasm. So they are intracellular. Whereas the ligand, hydrophilic ligand, hydrophilic ligand which cannot cross, for example, insulin which cannot cross this membrane, the receptor lies on the plasma membrane. Steroid hormone, for example, vitamin D, thyroxine, or testosterone, these hormone, they are lipophilic in nature, they can cross the plasma membrane, their receptor lies in the cytosol. Which class of hormone require a carrier protein? So the correct the this question is not complete. It is asking a carrier protein in the blood. So because steroid hormone, they are lipophilic in nature, they are hydrophobic in nature. So that's why and the nature of the plasma or blood is aqueous. So they cannot be carried in the blood as it is. So they require a carrier protein for their transport in the blood. Action of which class of hormone is mediated through a second messenger. So, mm, because hydrophilic, as I told, hydrophilic ligands, they cannot cross. This is the hydrophilic ligand. It can, because they are hydrophilic in nature, they cannot cross this membrane. So they interact through an through a plasma membrane receptor. So they interact with this extracellular domain of this receptor and then bring changes by generating a secondary messenger. So peptide and amine both are hydrophilic, both are hydrophilic ligand. So this class of hormone, they mediate their message through a secondary messenger like calcium cyclic AMP. Anti-fertilization is secreted by jelly, uterus, or kidney. It is secreted by the sperm, and the ova, it secretes fertilizing. So fertilizing and the sperm secrete anti-fertilizing. So there is a binding between there's an interaction between the fertilizing and fertilizing, which specify that only sperm of a particular species can fertilize the ova of that species only. So this specificity is provided by the fertilizing and fertilization reaction. Discoidal meroblastic cleavage takes place in Amphioxus, frog, insect, or hen. So correct answer is hen. So in case of hen, the yolk is telolacithal, and that's why there is a discoidal meroblastic cleavage takes place in hen. Metamorphosis in amphibians is controlled by estrogen, parathyroid hormone, gastrointestinal hormone, or thyroid hormone. So it is thyroid hormone which control metamorphosis in amphibians. So the next question is the regulation of limbs in salamander it is an example of epimorphosis. True. It involves differentiation of cells into blastema. True. It involves formation of an apical ectodermal cap from epidermis. True. So all of these options are right. So our correct answer will be D, all of these. Next question is alteration in the timing of development event is called as. Option is neoteny, B ontogeny, C phylogeny, or D heterochromy. The correct answer is heterochromy. Next question is torsion of body mass is observed at some time of development in Monoplacophora, B. Gastropoda, Pelecypoda, or Cephalopoda. So the correct answer is Gastropoda. Next question is main function of sweating is thermoregulation to excrete salt to maintain a blood volume or osmoregulation of body. So the correct answer is thermoregulation as well as to excrete salt. So both these options are correct. So please, for more such paper, please do share, please do subscribe our channel, and please share it with your friends and batchmates for whom it could be helpful. Thank you so much.